Thank you. It's good to be here. Magenta, this is the first time presenting at Cell and Gene, and look forward to bringing you up to speed. Where is my clicker? There we go. We're over that. That was our only technical problem we're going to have. We are a public company, and this is an important disclosure regarding forward-looking statements. At Magenta Therapeutics, we are focused on fundamentally changing the practice of stem cell transplant. As many of you in this room are aware, an immune reset through a stem cell transplant can be curative for that patient. But today, a stem cell transplant is still an option of last resort because of some of the dangers and the toxicities involved for those patients. And at Magenta, we're developing medicines to bring safe immune resets to patients with autoimmune disease, with genetic diseases, and for blood cancers. These are some of the faces of individuals who've received stem cell transplants. This has been happening for over 50 years, so about 1 million patients have received a stem cell transplant. These are a few. On upper left, this is Nancy, who 50 years ago had aplastic anemia and was cured from a transplant of cells from her sister, pictured next to her. The boys on the upper right both had leukemia and were cured. Jacob on the upper right through Magenta 456 product in phase 2 studies. Jennifer, shown on the bottom, had MS, and she received a stem cell transplant using her own cells, and she now lives life, in her words, without MS and without any MS drugs. And then Manny on the bottom is in a gene therapy stem cell transplant trial, and he's benefited now through transplant. So each of these stories is very powerful, and there's so many more like them. But it also reminds you that each patient had to go through this very difficult and toxic process. So what is that process? At a very high level, many of you in this room are aware of this, 2 major steps have to occur. The first is that stem cells need to be collected either from the patient themselves, from the bone marrow, or from a donor. Second, the patient needs to be prepared to then receive those cells. That is a very toxic process right now through conditioning. It's a very non-specific way of removing the stem and or immune cells in the bone marrow. Once the patient is prepared and the cells are ready, they go through a transplant. They receive those stem cells, they're infused, they return to the bone marrow, they go to the bone marrow, and they reconstitute the immune system. And bear in mind, there can be complications after transplant. So Magenta's portfolio looks to really change the way this process works for patients. This is, like I mentioned before, an option of last resort. You really don't want to today go through a transplant. We can fundamentally change that. Now, despite some of the risks involved in today's immune reset, we're seeing a rising tide here, first in autoimmune diseases. So over 2,000 MS patients have received a transplant with their own cells, and there is now data going out 10 to 12 years showing durable disease response, patients no longer on their MS medications. Likewise, in systemic sclerosis, which is a particularly devastating disease, it's got about a 50% 5-year survival rate. Patients who've been transplanted have shown significantly better and improved outcomes. And this same concept of immune reset can work in a number of other diseases. And yet today, this is still a very small portion of the patients who could benefit. They have to be willing to take that risk to go through with this and achieve that cure. Likewise, very germane to this conference, when we think of gene therapies and CAR T therapies, very powerful modern technologies. And all of these do rely on this backbone of chemotherapy to prepare the patient. Furthermore, in diseases such as sickle cell and these autoimmune diseases, it's very difficult with today's medicines to extract, to mobilize stem cells. So a number of different barriers. Across the US and Europe, on an annual basis today, there are about 65,000 stem cell transplants. These are mainly centered around blood cancers, used for blood cancer patients, but more so, as I just mentioned, around gene therapy, CAR T, and autoimmune diseases. That's growing, still small numbers. But notice there are 85,000 patients who are eligible who will not go through with the transplant. That can be because of the current conditioning regimen. It can be because they can't find a match. And it may be for a host of other reasons, including mobilization. Magenta very clearly wants to solve this equation, and we believe we can bring this safer immune reset to all of those eligible patients today, but also expand the size of this pie, MS, scleroderma, and a number of other diseases where patients should be able to benefit from a safe immune reset. And that's how we think of our program and our medicines. This is based on a background of stem cell biology and biotherapeutics discovery. There are 4 primary program areas for Magenta. The first is targeted conditioning, where we're focusing on antibody-drug conjugates to very specifically remove the cells that need to be removed from the bone marrow. Second area is mobilization. This is the flip side of the equation where you need those stem cells for the transplant, and I'll talk about that in a moment. We have a phase 1 study ongoing there. Third area, a cell therapy, focused in inherited metabolic diseases, I'll touch on in a moment. And our fourth area is in post-transplant complications. I won't go into much detail there today. So the number one barrier right now to a safe transplant is, in fact, patient preparation. The current medicines that are used are old. They weren't primarily designed for a transplant. They're non-selective and they're toxic, and they lead to, as you can see on the screen, organ failure, secondary cancers in a malignant setting. If it's non-malignant, it could be primary cancers, infertility, universal infertility, and worse. So as Magenta thought about what medicine could work, the business realized you need to be able to very specifically deplete stem cells and or immune cells and or disease-causing cells. And we're doing that with antibody-drug conjugates and using a lot of the knowledge that's come before us. And we've developed some of these. They need to be highly selective, they need to be very potent, and they need to be fast-acting. These have to have a short half-life because they need to do their job. This is a single dose, and then they need to clear the system because you need to make room for those incoming stem cells. Our first ADC program is Cd45 Target. And this is ideal for autoimmune diseases because Cd45 is expressed in the stem cells and on the immune cells. And obviously you need to remove the immune system for somebody with autoimmune disease. And we've seen encouraging preclinical data saying that this ADC does exactly what we need it to do, deplete the stem and the immune cells. We will have some data later this year, in particular at the American College of Rheumatology next month, in the first animal studies of autoimmune with this ADC, which is quite impactful. Our second antibody-drug conjugate targets Cd45. Now, this is expressed on the stem cells only, not on the immune cells. And this is ideal, therefore, for genetic diseases as well as certain blood cancers because you don't want to remove the immune system. In preclinical studies, we've shown this removal of stem cells and the maintenance of the immune system. We do anticipate additional data later this year. We've mentioned previously that we want to look at gene therapy models in non-human primates using a targeted CD117 antibody-drug conjugate. So moving along, the second program area, mobilization. Today, for all of those transplants that are happening, the 65,000, the vast majority of those are using cells that have been mobilized out of a patient or a donor's bone marrow. And there are a number of complications across different disease settings, multiple drugs being used, an average of 5 to 8 days to mobilize cells, again, out of a patient or out of a donor. And frankly, in sickle cell and autoimmune diseases, there is no good way to mobilize the patients. So we see a huge opportunity here, and the opportunity is to develop a one-time, same-day apheresis that can be reliable and predictable. And we think that by shortening this to a single day, a patient or donor walks in, receives the medicine, is apheresed, and you have the cells you need. That is a huge change, not only for scaling the system, but also for patients today who are suffering from the side effects of some of this mobilization. This is in a phase 1 study that we anticipate having data released later this year, likely around ASH. And this is exciting because we'll see not only safety and tolerability, but we'll actually see what stem cells get collected. And we know, roughly speaking, what we need to see in normal, healthy volunteers to equate to what patients will need. So that comes later this year. Like I mentioned, a significant opportunity across diseases. And finally, our cell therapy, Magenta 456, is in phase 2 studies right now. This is a large dose of well-matched stem cells. There is no genetic modification or genetic engineering involved. We are focused in inherited metabolic diseases, lysosomal storage disorders. And we've been very encouraged. We've said previously that we've enrolled 5 out of 12 patients, and we have 4 centers open. And we've seen with the Hurler patients and ALD patients very early engraftment as well as very consistent durable response. Because if you think about this, this is the equivalent of a vector copy number of 2.0. They're receiving cord cells that are well matched, significantly expanded, and we're seeing very little side effect by way of GVHD. So we're very excited about this, and it is a tailored, specific program for these IMDs, and it's a basket study. So I've mentioned going through a number of different catalysts. Q4 will be important for us. I think most importantly, we'll see a read-through on the phase 1 mobilization study. We will see data later in the year regarding C200 and non-humate. That's the CD117 antibody drug conjugate in non-human primates. And we'll have additional data we would anticipate for the cell therapy as well as autoimmune data for the Cd45 program, and lots to come next year. So in closing, we are quite passionate, as many of the companies are here at this conference, around the patients. And we think that with the medicines that are right now in our portfolio, it is absolutely a reality that someone like a Jennifer, who I mentioned at the beginning, who has MS, in the future could have her stem cells mobilized very easily in the same day, go through a completely non-toxic targeted removal of her stem cells and immune system, receive back her own cells, and go home without MS and without future MS drugs. This is happening today, but for very few patients who want to take the risk. We fundamentally believe in this future, and we're really passionate about that. So thank you for your time, and happy to take questions outside.